So this is acid and base titration. I'm going to turn off this light here. So very quickly, reviewing what we just said, we have that acids ionize in water to form hydrogen ions, but we just saw that it's actually more like a hydronium ion. Bases dissociate to form hydroxide ions. And when you have a reaction of an acid with a base, the hydrogen is transferred to the hydroxide to give me water, whereas the cation combines with the anion of the acid to give me a salt. All right, and that's why we say the reaction has been a neutralization, because we've eliminated these ions that we learned last time how to detect using indicators, litmus paper, all these different methods. Here is a table of some known acids and bases. And again, you can find this in your book. Again, you'll notice that most of the bases are hydroxides. Ammonia, as we saw, what it does is it generates with water ammonium and hydroxide ions. Some acids are strong, some are weak. A weak acid is one where the molecules don't completely ionize. Only a small percentage of them do. Strong acids, on the other hand, like HCl or sulfuric acid or nitric acid, you put them in water, every single hydrogen just dissociates out or ionizes out. Okay? As I said, when you are doing a chemical reaction, we have studied stoichiometry, first of all, with pure substances, where, let's say, for example, my reactant in question was given in grams. And to bring it into the chemical equation, I had to convert it to moles. So I could use the ratios of the reaction to calculate either the other reactant or the product or whatever. When we studied reactions with gases, we know that because moles and volume are related. I'm sorry, when we moles and volume and pressure are related, we can use the ideal gas equation to calculate the number of moles at a given pressure, volume, and temperature. With solutions, we typically have a molarity, and what we add is a certain volume of one solution, and knowing the molarity as a conversion factor, we can calculate how many moles we added. So that's essentially how we're going to do this. The apparatus we're going to use is called a burette, and the technique is called a titration. So in a titration, what you do is you react to solutions. One of them, you know the exact number of moles that are in there. The other one has the other reactant in unknown concentration. So knowing how much reacted of the one you know, you can calculate how much of the other guy there was and then calculate its concentration. We're going to use an instrument called a burette. And a burette is a long glass tube, and I have one here. It has a graduated scale on the side here, and it's calibrated in tenths of milliliters, which means when you record a measurement on it, you have to report it to one more decimal, right? It also has a valve here that you can use to open it or close it, all right? So that's the setup of the burette. Now, the substance that you put in the burette is going to be called the titrant. The titrant might be your known or your unknown. And then you're going to drip that liquid into a reaction beaker or flask that has the other reactant. When they finish reacting, at the point where they are in the exact ratios that the chemical equation gives you for a complete reaction, we call that the equivalence point. The equivalence point is the point at which every single unit of reactants has reacted. However, it turns out that for acid-base reactions, there is no visible indication that the reaction is finished or not. So we need some help. What we do is we add something called an indicator. So it's a chemical that will change color when the solution undergoes a large change in the level of acid. So at the equivalence point, what's going to happen is the number of moles of hydrogen ions is going to equal the number of moles of hydroxide ions. That's the equivalence point. We can't visualize that. But what we do is we put an indicator that will change color as soon as one of these two exceeds the other one. All right? And once the conditions are off balance, the indicator changes color. And we use that, called the endpoint, to approximate what the equivalence point was, okay? The idea is that if you do it right, they are so close to each other that within the typical percent error in all these experiments, you're gonna be very close. And you can calculate molarities to 4 decimal places using this method, all right? So let's talk about the use of the burette. Okay, as you can see, you're gonna mount the burette on a ring stand, and the ring stand is gonna have a special kind of clamp that's called a burette clamp, and I have here a sample of it. Notice that it's a pressure clamp, and these clamps are located at the end of this table here in a box in there. Of course, the ring stands are located in that other corner of the lab, all right? And the burette is going to be mounted in here using the pressure clamp, all right? So I have it in here, and I'm gonna show you in a moment. I'm gonna move it over here so you can see it better. The reaction requires that you observe a very faint color change. For that purpose, you typically want to put your reaction flask over a piece of white paper just to help with your visibility. All right? So just to help with your visibility. Let me put this a little here. Okay, there we go. The other thing is that when you read the burette, of course, like any other, long tube, narrow tube that has water in it, it forms a meniscus. So the way you read it is you're going to read it at the bottom of the meniscus. All right? And notice that because these intervals are tenths of milliliters, you have to report it to the hundreds. In other words, to 2 decimal places. All right? Okay, so let's talk about, first of all, how we're going to set this up. Okay, let me, first of all, let me blank this out for a moment. And let's come back here and show you how we're going to do this. All right. So today, if you look at your lab manual, you're going to use an acid standard. In other words, this is an acid that is very stable, and it allows you to calculate a very exact number of moles of it. When you wrap this up, of course, you're going to go get a test tube. They're over there by the window. And in each test tube, there is this substance. It's called potassium hydrogen phthalate. And you have the word in there in your lab manual, right? Part B. We abbreviate it as KHP. But remember, KHP is not a formula. It's not potassium hydrogen phosphorus. It's a nickname. It's an abbreviation, all right? KHP is a solid of large molar mass, which makes it very stable. I think its molar mass is given there 204.22 grams per mole, something like that. And in each one of these test tubes, you're going to have enough for 2 trials, all right? So when you bring it back to your bench, what you want to do is grab another test tube and visually kind of divide this into 2 portions. Kind of like half and half, a little more here. And again, like I said, you can pretty much eyeball it. There should be roughly about 1 gram in each one of these samples, okay? So let's start with one of them. I'm going to save one for now. And with the other one, what I want to do is I want to dissolve it in water. So I'm going to grab my 250 ml flask. I'm going to put it on the balance, and I'm going to zero it or tare it, because I want to measure exactly the amount of acid that is in here. All right. So I'm going to zero it on the balance, and then I'm going to pour this in, and then I'm going to record the mass that's in there. Notice that because I zeroed out the flask, the mass I'm recording is exactly the mass of KHP that's in the flask. All right. Now what I want to do is I want to dissolve it in water. Every time you mix chemicals and it says water, it means distilled water. So I'm going to put in approximately 50 milliliters. If your flask has markings on the side, then you should be able to just eyeball it. So I'm going to go on here and I'm going to kind of like swirl it, kind of shoot it down the sides just to grab everything that's in there. Like it says there, it's approximately 50 milliliters. It doesn't have to be exact. So I'm going to put approximately 50 milliliters, and then I'm going to simply swirl it to dissolve the acid. Now here's a situation, and it is that sometimes these acids are not very soluble. And by the way, I should be wearing my goggles at this point. All right. So if that is the case, what you want to do is, and I don't have my Bunsen burner here for some reason, what you can do is you need to make yourself a like a collar with paper towels, and paper towels are gone too. Yeah, I was looking for the brown one. That's a So here's one way you can do it, is you can take this and kind of fold it several times, and then kind of like wrap it around the neck of your flask and twist it tight. And that'll give you like a little collar to hold your flask. And then what you do is you turn on your Bunsen burner. I don't know what happened to mine. Very gently, kind of let it kind of like float over the flame. Don't get it too hot. You don't want it to boil. But the heat helps dissolve the acid. And once it's fully dissolved, bring it back and just let it kind of cool down a little bit back to room temperature. All right? At this point, we need to put an indicator. Let me look for the indicator. It's over here by the window. So the indicator is in a little dropper bottle. So what I'm going to do is I'm going to grab maybe like a couple of, a little bit of it, and then squirt maybe a couple of drops in my reaction mixture, and then kind of like swirl around, mix it. All right. And then probably because I was swirling, I want to make sure I get all the acid in there. So I'm going to squirt it. Notice that I don't really care how much water is in here. What I care is how much acid was in there. And I get that from the grams that I weighed it, which I can convert it into moles. Okay. So that is my reaction mixture. All right. And I'm going to put it aside for now. And let me show you what we just did here. Just a review here. Okay, so first we obtained the sample of KHP, which is potassium hydrogen phthalate. Then we tared or zeroed a flask. We took about half of the sample that we had and we measured it. Should be about a gram. We added about 50 milliliters of distilled water. We used the washcloth to make sure we rinse around the sides to make sure we get all the acid solid inside the bottom, in the bottom of the flask. We dissolve by swirling. If we needed to warm it up with a Bunsen burner, that's fine, but then bring it back to room temperature. Add 2 to 3 drops of the indicator, which is the same one that we used last week, the phenolphthalein. Remember, that's the one that turns pink when you put it in base, which means that right now it should be colorless. If it turns pink, there's something wrong with your preparation. You got it contaminated or something happened in the process, okay? Now, the next thing we're going to do is we're going to prepare or condition the burette. So when you retrieve the burette from the window there, it may have a few drops of water in it from when it was rinsed. So we need to condition it. We have to make sure that the only thing that's in here is our solution which is going to be our KOH. So what you're going to do is you're going to go to the shelf over here, and you're going to grab your bottle of KOH that you prepared last time. Remember, we at the time figured this is approximately 0.5 molar, but we want to know exactly what it is. So we're going to use this as our titrant on the burette and react it with our mixture of acid, which we know exactly how many moles are in there, to find out how many moles are in a sample of this. Now, it's hard to pour from here, so I would suggest you grab a small beaker and take some of this and put it in there. Maybe, I don't know, maybe like 100 milliliters of this. The reason being that with the beaker spout, you can pour more easily from it, all right? Then what we're gonna do is, you're gonna come and get your burette. Make sure that the valve is closed, which is when it's perpendicular to the body of the burette. And you're gonna take some of this, about 5 or 10 milliliters of the KOH, and pour it down very carefully. All right? And then what you'll do is very carefully turn it on its side and then start rolling it so that the whole interior of the burette gets essentially rinsed with your KOH. All right? And then bring it back. You should have like a beaker for waste and stuff, and just let it empty into that. That way also the tip of the burette will also kind of clear out and get conditioned. I think the protocol maybe tells you to do this twice, but I think one time should be enough. This also allows you to check that it's flowing well, okay? And then let it flow all the way out, and once it's out, close the valve, and then you're gonna bring it back to your stand, all right? So I'm gonna put it back on its stand here. And let me move this stuff out of the way so I can roll it over here. I'm recording this, so hopefully people are getting to see what's going on when they watch the video. So here's my burette. It's in here. Now, let me go back to our presentation before I do that. All right, come back in here. Let's see. So basically, make sure the burette stopcock is closed, 90 degrees from the body of the burette. Add about 5 to 10 milliliters of your KOH solution. Then tilt the burette to an almost horizontal position, rotate it to rinse the interior. This is what's called conditioning the burette. It says here to repeat it. I don't think you need to repeat it. One time should be enough, all right? Then put it back in there and open the stopcock to make sure that the whole thing flows out and there are no trapped bubbles and the liquid flows smoothly, all right? Now we're gonna fill it up with KOH, and we're going to let it drain a little bit to bring the volume marker to about 0.00, and then wipe the outside with paper towel, all right? So here is my burette that I just prepped, all right? Again, now I'm going to fill it up. This time, I'm going to use a funnel, a glass funnel, to fill it up. All right. Let me pull this up here. I'm going to use a glass funnel. And again, I'm going to pour from my beaker. Now, be careful. Sometimes the glass funnel makes a seal here. And then when you start pouring the liquid, the air pressure inside builds up and doesn't let any more flow. And then you're going to have a spill. So maybe hold it a little bit higher and then pour it down. And it'll take a little over 50 milliliters of your KOH to fill the burette. You want to bring it a little higher than the 0 mark. All right. And then pull it out. Don't throw away your KOH, because we're going to need it for a future experiment. Well, assuming we get to do it. Right now, it doesn't look like we will, but I want you to keep it anyway. Now, remember that you have to read the burette at eye level, which means that you may have to kind of lower it a little bit if you're vertically challenged like me. right? Put your waste beaker underneath, all right? And then there are 3 ways that you can allow a burette to flow, okay? One way is to completely open the valve and let it stream out. Another one is to open it slightly, very carefully, and let it drip slowly. Let it drip slowly, all right? And the other one is, because water has this property where the molecules hold on to each other, when you simply let gravity pull a stream of water down, it holds on and it forms a big drop. If you form a big drop, that means that the amount you're delivering might be a little too much for what you're trying to accomplish. So to get a micro drop, what you do is you do a very quick 180 turn of the valve, and what that does is it squirts out a micro drop. That way you can control it a little better. So right now I am at about a little below the 0 line. It doesn't matter where you start or where you finish, because the burette is not meant to measure what volume of liquid is in it. It's meant to help you decide how much liquid was delivered into your reaction flask. See what I'm saying? So it starts at 0 on the top and it moves towards 50. So what you record is your initial and your final. If you can get to start at 0.00 milliliters, that's fine. I wasn't able to get it because I was demonstrating these things for you. So right now, my burette, when I read it, is actually at 0. Oh, you know what you can do if you're having a hard time seeing it, is put a piece of paper kind of behind it. And that way you can kind of better read what's in there. So right now I am at 0.20 milliliters, okay? 0.20. Okay, now I'm gonna bring it back up. I'm gonna take a clean piece of paper towel and kind of give it a little wipe on the outside, make sure that nothing's spilled, because I want the only liquid that I want in my reaction mixture is the one that's inside the burette. I'm gonna bring out my reaction mixture here, and because I'm looking for a very faint color change, I'm going to put a piece of white paper or white paper towel underneath so that I can watch it. All right, I'm ready to start. I have my reaction mixture down here. I have my burette with my unknown KOH in here, and I'm going to start now changing. Now remember, the idea is that as the KOH reacts with the acid, it starts neutralizing. At some point, the acid is going to be so far reacted that the base is going to start reacting with the indicator instead. And the indicator is going to start changing color. But because there's still so little of it, it's going to be like a faint color and then disappears. So as I go through, I'm going to watch these swirls of pink color form and disappear, form and disappear. The way you do it, you start with a slow dripping kind of delivery. So I'm going to start by opening it slightly here and letting my titrant fall down drop by drop. As it drips down, okay, I don't know if people can see it here on the picture. Oops, sorry, wrong one. Let's put a piece of paper here. No, you can't see it there. Anyway, whatever. The point is that it is dripping there slowly. And as it drips, I want to be swirling this constantly, all right, all the time. I want to make sure that I can see the color change. As I swirl it, I'm going to see swirls of pink forming and disappearing. When those pink swirls start kind of lingering for about 5 seconds or so, I want to stop. At this point, I want to make sure that I've captured everything that was delivered in. And since I was swirling, there may be little drops of KOH on the walls of my reaction flask. So I want to rinse them down with my squirt bottle of distilled water. Swirl it some more. And now from here on out, I am going to be doing the 180 turns. Swirl, 180, swirl, 180, swirl, all right? And the idea is I'm being very careful because the closer I get to the, the equivalence point, the harder it is going to be to keep it from overstepping that point, all right? What I'm looking for is for a very faint, almost imperceptible pink color that lasts, that lingers for about 20 seconds. Once I get there, I'm going to, again, rinse out my flask, swirl it some more, okay? I'm going to check what the volume is at that point. I'm going to write that down as my final volume, all right? And now I'm ready. If I want to be sure, I might want to give it like one more shot. Okay, I already wrote that down. One more shot. And what will probably happen is that it'll overshoot and turn neon pink. That means that the volume you recorded was the right one, all right? You don't have to refill the burette. You can now take the volume that's in there and count that as your initial volume of your second trial. And for your second trial, you prepare another reaction flask with, the rest of your KHP, same as you did here, and repeat the titration, all right? Now, your goal is for 2 titrations to be within about 5% of each other in the final result, all right? If you overshoot one, if it goes to neon pink, just call it off and just do another one. But you can only do a maximum of 3. Okay, that's the most you can do, all right? Let's go back here to our PowerPoint. I'm going to move this out of the way, all right? So I can tell you what's going to happen next. And then I'm going to walk you through a sample set of calculations. So here we go. Like we said, hold on a second. Let's back up here. There we go. You're going to record the initial volume in 2 decimal places. While swirling the flask one hand, open the burette valve slightly to obtain a drop-wise flow of the KOH. That's just kind of drip, 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 drip. Okay? You observe these swirls of pink color, and as soon as you see one that lasts for about 5 seconds, stop. Close the valve. Rinse the interior of the flask with distilled water. Make sure that every single drop of KOH was actually delivered to the reaction. All right? Now you continue using the 180 turns. Guys, all this is written in your lab manual. You don't have to write it down. And when you see a very faint pink color that persists for more than 20 seconds, that means you've reached the end point. All right? At this point, read what the final volume is. And this volume is going to be the initial one for your second trial. Okay? Here's the pictures of what happens. Here is a sample of acid being titrated with NaOH. You can see the swirls of pink that happen as it goes through. And this is your endpoint, a very faint pink color. Let me show you some other set of pictures you can see. This is, as I had a student do this for me. So you see the one on the left? That's not good. That's an overshot titration. See the one in the middle with the obvious pink or the light? That one's so-so. See the one on the right, almost imperceptible? That's beautiful. That's what you're shooting for. A very, very faint, almost imperceptible color. And, if you find it and then you come and say, Dr. Diaz, come and see my result. By the time I get there, it might have disappeared. It might be colorless again. That's a good titration. That's what we're looking for. All right? It's that faint and that short-lived. Well, it should last about 20 to 30 seconds. All right? At least. What happens is the carbon dioxide in the air makes the solution acidic. So it turns the indicator back to colorless by being there.